What I'm here to talk about now is my research expertise, which, as I said, I'm trained as a statistician, and I was hired at the University of California to be a dairy cattle geneticist, and I've worked with beef and pigs and shrimp and other agricultural species. But for the past 10, 15 years, I've also been doing some work with dogs. And so I'm going to talk a little bit about some of the work that I've done with dogs that may be applicable to some of you in the room that are coming from breeds that are rather small populations and had to confront issues of withering genetic diversity that is increasing inbreeding, and yet you still want to do something with the animals, take them in some direction, run some sort of a breeding program, if I stress the right thing. So the goal of today's talk, as you can see, is to address the concerns of private breeders and ask the simple question, is it possible to maintain genetic diversity in populations of small size? And if so, are these strategies realistic? And what are the challenges that one will be confronted in trying to run these strategies? Because what you're going to find out, I think, not to give up the end of the talk, as exciting as it is, I know everybody still wants to have lunch, so maybe I give you the high points now. We'll find out that there's some very elegant mathematical solutions to these problems, and people like myself like to sit all day in my office and drink coffee and write on a pad and come up with an elegant answer. But putting that into a practical program that people can actually use day to day, that will be one of the challenges that we're going to have to talk about. But again, keeping in mind that our goal here is to help us better manage the animals that we hope to care for. For most of the breeds that we're talking about, again, this is not so much to tell you. I think you already know what your breeding goals are. Again, I have to walk away from my dairy cattle background and remember that we're not breeding for production traits of some sort, the lean body mass or something of that nature and size of steaks. So I'm going to remind myself that in your case, we're interested usually in minimizing risk to disease. One of the major focuses of any breed club is some element of genetic or inherited diseases. At the same time, minimizing the accumulation of inbreeding, particularly in these small populations, and to be able to accomplish this without sacrificing any of the identifying characteristics or distinctions that your breed is noteworthy for. And again, to remind myself, really the first project I worked on with dogs, one of the students in my class, her mom was a veterinarian who did bear testing, the hearing testing in Dalmatians, and so that was my introduction to dog disease genetics was through Dalmatians. I put them up there just to use them as an example of a situation where we all know quite well whether we can find the exact genes responsible for deafness in Dalmatians. We understand that it's clearly related to the coat color patterns that we've selected. What makes a Dalmatian such a cherished animal is its coat color pattern, or at least one of its many myriad of elements that people love. But if we were to try and remove deafness entirely, we'd probably wind up with a dog that doesn't have any more spots. And that's really what I want to get across with that sentence of we want to be able to maintain the populations for what we want to characterize them as, but not give up their essence and why they are such a cherished breed. And that is the essence of so many of these breeding problems. We think of solving the problem with a selection program, and yet in the end, we may come back with an animal that's not as desirable as when we started. So we have to keep that in mind as we go through these programs. Our concerns about genetic diversity are only natural. Whenever I mention to a class of freshmen in my animal science classes that I do a little bit of canine disease genetics on the side, it's almost immediately, people will just say, well, that's because of inbreeding. That's exactly why. Just inbreeding makes disease. And of course, we should know in this room that inbreeding does no such thing. It doesn't create disease. It's a useful tool for any animal breeder. There are times when inbreeding is absolutely essential. And in fact, most of the food we eat around we'll be having for lunch is going to have some element of inbreeding involved in its production. So it's a useful tool, but it's one that has to be used wisely. And we also know as breeders that it's inevitable. And inbreeding is always going to occur. It's a natural phenomenon that you cannot avoid. And if we go back, I'm not going to have any, well, I can't say I'm not going to have any equations. I'm just going to have some, but they're not really bad. There won't be an exam or any such thing. But, we all understand that in a population genetics class, we would talk about this accumulation of inbreeding that is just natural and part of any closed population. Well, again, to remind us, though, of how inbreeding manifests itself in a breeding program, regardless of the breed you're working with, I always like to remind myself, again, of the basic axioms of animal breeding, which we don't need to be reminded of: breeding the best to the best, and like begets like. Again, I leave those, I bring those to remind myself. These are the quotes of the father of animal breeding from 1740, of Robert Bakewell. This is not a new process. This is not something we all understand how animal breeding works. You don't have to go to a university to figure it out. And we could have seen that in our yesterday's talks with our 35,000-year-old dog Bones. They were doing animal breeding too. They understood that you could breed the best of the best and like begets like. They may not have understood all the forces or the mathematics behind it, but they certainly had that essence. And it's not really rocket science in that regard. At any rate, so inbreeding is something we usually want to avoid, but I'll give you definitions. Of course, you know, what is inbreeding? We know that, we should all know that it's the mating of relatives. The pictures on the side, I feel really self-conscious. I don't work directly with animals in my research. I spend all day with computers and pads of paper and just drinking coffee. And other people have such nice slides of dogs that are cute, or animals, and they got videos. So I was thinking, well, for the talk, you know, I could Google. Don't do this. Google images for inbreeding. You don't want that. You just don't want. But that was. But I did manage to find. Okay, that's the the royal family of Spain from some hundreds of years ago. You know, we all know what inbreeding is, mating of relatives. There are different ways to describe it. The formula there for populations, for those of you that may have had this in a genetics class at some point, is really measuring the concept of heterozygosity. H means heterozygosity. That is. We all know that you get one allele from your mom and one allele from your dad, and if they're the same, it's called homozygous, and if they're different, they're heterozygous. And heterozygous is just, you've got this mixing of genes, and homozygosity is the exact opposite. And inbreeding is just this accumulation of homozygosity that occurs from the mating of relatives. We can talk about calculating it. The statistic for inbreeding is F. It was named by a self-centered statistician, very famous one. He got to be a Sir out of it, but Sir Ronald Fisher, he studied inbreeding for the first scientist to really uncover the properties of this process, and so, not being bashful, named it after himself and used F as the symbol. But as the formula indicates, what inbreeding does is that over time it decreases heterozygosity and increases homozygosity. That's all it does. It doesn't make disease, but it does expose disease. As I have in the bottom, that bad recessive alleles, when they pair up, that increasing in homozygosity creates or manifest. We see the disease. They're always under there. Those bad genes are always floating around. We'll never get rid of them. But inbreeding does expose them and lets us see those diseases. And sometimes that's good, sometimes that's useful. We can get those animals out of the population. So it can serve a useful purpose, but it is the cost of doing business when mating relatives. Historically, then, and this was mentioned a little bit yesterday as well, I think Scott brought it up, and that it's relatively simple to measure with some computer programs. I won't go into the details today, but that historically it's very easy to measure. You just need a pedigree. You need to know an animal and who his parents are, and know that for as far back as you'd care to go, and can calculate that. There is a growing, if Professor Wayne were here today, though, he could talk about another way of measuring estimate of homozygosity and hence inbreeding is by actually looking at these SNPs and microsatellites. So we now have two tools, whereas in the past, when I was in school, we learned to calculate inbreeding strictly from pedigree information. Now, with genomic information, we could calculate it in that pathway. You'll get different answers. They're not in close agreement sometimes. Usually they are. But they are still measuring the same phenomenon in the animals. Increases in homozygosity brought about by the mating of relatives. You can sing that at lunch today. I'll say that so many times. Increases. Oh, well. Okay. Sorry. I don't have to go into any great detail. I could do dozens of slides that indicate what inbreeding depression is, but it's, again, that phenomenon that my students were talking about when they said that inbreeding causes disease. Not exactly, but very close. And this is just an example. You can't really read the numbers too well, but you get the general sense that it's going up, and this is the rate of inbreeding on the bottom and the fraction of the population that has some form of hip dysplasia. These are Icelandic sheepdogs. I could have found any number of examples, so I don't mean to blaspheme any one particular breed or not. but just to give an example of, we know that this is the occurrence, and this is in fact what we're trying to avoid. So as we think of our goals for running a breeding program, that we're not just simply, usually not trying to just have stasis. We don't want to just keep the population the same. We're Americans. We're here for change. We always want new things. We want better things. And so we always have some idea of improvement in the future. And as I say here, in our situation for dogs, we're often talking about decreasing the incidence of disease, perhaps improving some element of behavior or temperament traits that we're concerned with in our breed or in our own animals, or addressing some structural or conformation problem. The basic information, we have something we'd like to change. So we have this small, closed population. We don't want it to just stay still. We do want to move it in a direction and take it into a better place, ideally. And the ideal method for making those decisions, again, this was a word that came up yesterday in our talk about the dogs at TSA, was the ideal method for doing this is something called estimated breeding values. That was what, those equations on the previous slide, they comfort me. And so I looked at, they remind me, we're going to talk about estimated breeding values. It's a very elaborate statistical technique, but what we're really hoping to do is take the phenotypes we see, marry that with the pedigree information we've collected, and try to look exactly into the genome. Professor Wayne is trying to do that with the actual DNA. It's a process that has not met with great success in livestock. I don't see the day when it will work as well in dogs either. It's something that's why it's still a research tool, to be able to look at actual chunks of DNA and say, this is the phenotype we expect. This is the value of this animal. This is the one we should breed, and this is the one we shouldn't. We're still left with largely statistical tools. They've worked quite well. They're widespread. They have widespread use in livestock. All of the dairy cattle, beef cattle, pigs, major species of livestock are all bred under a system of estimated breeding values, run either by the United States Department of Agriculture or various breed societies. The problem is that it has limited application in dogs, the places, because you're not essentially a cohesive unit or business that all work together. The few places that it works with dogs is in colonies, the rather larger colonies of service dogs, which is, and I'll bring up guide dogs for the blind as an example in just a bit, for whom I serve as a consultant. But in a closed colony, just try and picture this though. I'm going to try and evaluate who are the best animals in my closed colony of dogs. I'm going to predict their genetic value, this estimated breeding value. It's logical, however, they're going to wind up being correlated to one another. We know that records on relatives are correlated, hence their genetic merit will be correlated. And one of the problems of using EBVs is when you're using them to identify superior individuals, the animals that rise to the top, that is, those with the best breeding values, are more than likely going to be related to one another, which is going to exacerbate any increase in inbreeding. So that if our increase in inbreeding. So we get two reasons that our inbreeding coefficients go up in our small populations. One, because it's a closed and small population, and no matter whom you mate with, even if you chose them at random, it's likely they're going to be a distant relative at least. But when we want to run a breeding program and select the best, and identify those best animals and select those, we're exacerbating that inbreeding issue. We're more likely to put in our pot of the best individuals that are related. And so selection poses this additional problem for us to solve if we're trying to maximize diversity, minimize inbreeding. So the question that's really necessary to ask then, and getting down to the brass tacks of this talk, is it feasible? Can one maximize the genetic merit of those individuals who are going to be the parents, which is the goal of a breeding program, and at the same time constrain the increase in inbreeding? Can we, these two things are fighting with each other, can we find a stable answer? And that's where mathematics comes in and is very handy. That is, yes, we can do that. It's not simple, but it certainly is feasible. And this is just one of several papers that deal with that topic in the Journal of Animal Science, of course, of all places, where we would worry about this less with dogs, but more with livestock. This same issue of small breeds and small populations of animals in which we want to select the best, but similarly have to confine our inbreeding. And so in this particular paper, it goes through great details of mathematics. If you like calculus and vector calculus, it's fantastic. Have a great time. I almost was going to type these things in, but it was too much work and I was lazy. But just to give you the flavor that it's not going to be a simple process. It is feasible. We can maximize genetic gain in the next generation and minimize the increase of inbreeding in the next generation. But it's not something you just routinely pull off the shelf and is something simple to do. So that given that the arithmetic necessary to do this is it's unlikely you're going to ever see this implemented in practice. It's something very often is just one of these papers and one of these processes I'm as guilty as the next scientist of finding out some new information and writing papers and working very hard on a research topic, knowing full well it's probably never going to ever be put into use. It's just too challenging until you can find a simpler way to do it. And that's even in a colony, we'll say like Guide Dogs for the Blind, where they've invested some resources into making wise breeding decisions using EBVs. Even for them, it's a very challenging problem to then go ahead amongst all the animals that they've decided to select as parents, how they're going to use this mathematics algorithm to decide how to mate them thereafter. Well, we'll get into some actual information then and try and give you some idea of what I do for Guide Dogs for the Blind every month. Because to show that even though the math is really challenging and no one's going to be able to put it in place as it's been written in the papers, there's some ways to play with this and just using some straightforward logic and intuition. And that's what seems to be going on at Guide Dogs for the Blind. As Scott was saying yesterday, you make up a numbering system of 1, 2, 3, and then somebody throws in a 2.5. I'm sure it means a lot to you, but it can drive a statistician crazy. And not that you need to worry about my sanity, but nonetheless, these are very simple. They're yes, no. The dog has cataracts or doesn't. The dog is fearful or not. The dog has tricuspid valve dysplasia or not. And that's the simplest way we do this. And then they send me the data every month. I do a series of calculations, and then send it back to them with the hope that they'll use those numbers to decide which animals to use as breeding stock and which animals that they will send out to the visually impaired. So to give you an idea, again, the basic structure of Guide Dogs for the Blind in San Rafael, they've come up in several of the conversations these past 2 days. It was a service dog nonprofit organization that was developed shortly after the Second World War for returning veterans from the Second World War. In other words, their clientele in the onset and their mindset for the clientele of individuals when they first started were active young men. Individuals who were going to go out and still wanted to lead active lives, and that's the kind of dog they were going to have to provide for them. Of course, today their clientele is a lot more diverse. There are still active young people, and there are also people that are not interested in being active. They've lost their eyesight perhaps from other diseases like diabetes. It's a very different market for that dog. Their breeding strategies have got to encompass a variety of niches, whether it's a dog that wants to go outside perhaps and work on a city street versus a dog that would rather just sit around the house most of the day and hang out, answer the phone, and so on. So, their emphasis, though, of course, is on health and temperament traits. The basic numbers for their population, again, summarizing this in rough numbers, they produce about 1,000 puppies a year, and they do the process where they whelp those at their own facilities, and then they're sent out for rearing families. About 800 dogs will come back to the rearing facility. Of those 1,000, we'll get about 800 puppies back, dogs back at 14 months of age when they'll come back for their training. The loss between 1,000 to 800 dogs, again, the predominance is behavioral problems with some elements of health problems. In fact, we can look at some of the other numbers that again, very crude numbers I can take out of the data set for any given year, that of the 800 that return for training, the great majority are lost for some element of a behavior problem. The most common behavior problem, and this has come up in our discussions earlier today, is something like a distraction. A dog that's is supposed to be paying attention, and he wants to go, instead of chasing his friends or some smell or aroma, he'd like to do, and he's really supposed to help this visually impaired individual. So those sorts of behavior issues come up. Health problems, they can range from, as we've seen, heart disease to some cataract issues to allergies. I was thinking of the others. Of this, 350, 400 dogs, again, roughly vacillates from year to year, graduate. That is, they're good enough to be placed with a visually impaired individual. And the purpose behind my giving them these estimated breeding values is to find the 50 or 60 dogs each year that they'd like to keep as breeders. So those numbers are not really to determine anyone's going to be placed with a blind person. That's something their organization determines. It's rather to use those numbers in concert with their own opinions about the dogs, because this is not a strict mathematics problem where they just take the numbers and go blindly. It's using the numbers and their knowledge of the animals to decide which will be set aside and used as breeders in the next generation. So to give you, without worrying about the scales or what these numbers are really intended to be, what I wanted to give you is one of the values of using these statistics is I can tell you what's happening to your population over time. Going back to 1977, I can look at the genetic value for all of these traits that I predicted with my equations and average them by year of birth. And so, for example, what we have here is the average genetic merit from 1979 to the year 2010 for, in this case, of cataracts. And the way we score the trait, as I said, was a dog is normal, is 0, and a dog that has cataracts is a 1, so bigger numbers are bad. What we'd like to see is a nice steep trend in decline, that they're running a really good selection program and it's moving down. As you can see on their cataracts trait, they're not as successful as I think they'd like to be. I take no responsibility for this because I didn't start working with them until about the year 2000. And so what had happened obviously is they didn't even collect cataracts data here, but just to give you an idea, they saw a problem coming up around the year 2000. They contacted me for, the person who runs their breeding program is a former student at Davis and who knew of my interest in statistics, so I asked if I would help. But as I said, they probably would like a steeper decline in that number, but the basic idea is we'd like to see it dropping. That would be saying that the risk of getting cataracts is declining in our colony of dogs. Here on the case of TVD, on tricuspid valve dysplasia, the same scoring system. A good dog is a 0, a bad dog is a 1, so we would like to see it dropping. Lower values would be better. And here you can see very nicely, particularly since the year 2000, a really nice steady drop. They're doing a very nice job of being able to choose among those individuals that are not at risk for this heart disorder. In other words, what they're doing, closed colony, that increases inbreeding, making selection decisions, that increases inbreeding. But we are at least seeing that they're getting some benefit from it. A little bit of drop in cataracts, nice drop in TVD. I could give you other traits. Here's the one on fear. Again, scored, a dog is normal is 0. The score for 1, I don't know how they, by the way, they come up with this measurement of fear. That they don't tell me, that's a secret. But they just tell me this is a fearful dog, and I dutifully record it and put it into the computer. But again, we have this notion where a 1 is a dog that's fearful and has been removed from the breeding pool, it was released because it's a fearful dog. And again, dropping. I could go on in this for several other traits. I figured 3 would give you the basic message that they're making selection decisions and they're making progress. That should have, the downside of that should be an accumulation of inbreeding. And that's the really good news that comes out of this. This is looking at the average inbreeding coefficient of their colony, which has this, because of the program running going back to 1977, we get a nice increase. But since the year 2000, when we started playing with these numbers, it's flat. Now it's at 8%, and the discussion yesterday was around 3%. And I think 3% would be great, but I don't think we're going to get there. Certainly not in this colony. It's going to be, we have to bring in new stock. It's always going to go up. It's always going to accumulate. But what we're able to do here, as you can see, it's relatively flat. They have a closed colony. They're making breeding decisions, but it's not going up. It will eventually slowly, but in other words, they're able to damp down the increase in inbreeding, i.e. maintaining their genetic diversity. How do they do that is, well, I'm saying they use roughly 30 sires. I put up there, they have 30 sires and 100 dams each year and so on. But really, all they're doing is they're not using the math. They're doing what would be logical. They're looking at the individuals who could be mated, and they're determining, are they related to one another? So they just consciously avoid close matings. They don't have to get out a computer program and go through this fancy algorithm that is derived in these other papers. They're just simply making what would be an intuitive or logical decision. Here are the individuals that could be parents. I'm going to make them accordingly so that I've got them as far apart as possible. And that's the tool they're using. So to get us down to the final answer, in other words, is it possible to build a breeding program that improves performance without limiting diversity? And then we can see that it is indeed at least feasible. It's doable. I'm not sure that it's simple to do. It's certainly for all of us in this room, that for the particular breeds in which you come from. But in the case of a closed colony and an organization like Guide Dogs for the Blind or Canine Companions for Independence that Paul runs, they at least have the resources. The resources that being that they can go out and collect phenotypic information, so they can go out and measure these animals in repeated strategies, and they all agree on the measurement process. They can keep track of their genetic information, that is their pedigrees, and because there's really only one or two individuals involved in this breeding process, I don't know how many people make decisions for you, Paul, but it's probably just you. You get somebody else gives you advice or pokes you with a stick. Well, that's good. But I was thinking it's one person who's making that decision, so it's a little easier to be a dictator then. And so you can make that commitment over a long period of time, because as we know, animal breeding is a slow process. It takes a long time to see the results of these. You don't get immediate rewards. So as I close, to remind you of what you can already see are your challenges, is that the first thing everyone has to be able to agree on is that you can measure the traits that you're interested in, whatever your breed club would be focusing on, in a repeatable and accurate manner that everyone can agree on. And that can be a real challenge, particularly for some of these disease traits. I had to beg for them to give me the tricuspid valve dysplasia scores as a yes and a no, because their cardiologists couldn't agree. All looking at the same data, we'd have no's, suspects, and yeses. And we had to make commitments. I can put suspects, it can work that way, but I really, what we've got to, but I use it as an illustration of, that's not, just because I say it's, oh, we'll get easy, repeatable measures, it's easy to say, it's hard to do sometimes, particularly for behavior traits. We just saw that, we discussed that a little bit yesterday, and I brought it up again. We want to have 3 categories, 1, 2, and 3. Oh, I think that's a 2.5. Well, now we're out of luck again. So first of all, you've got to agree on how you're going to measure this trait, because you're never going to make it change if you can't agree on how to measure it. Everyone has to agree to work to produce good pedigree information. Again, I know that is what you're trying to do. No one's trying to screw up on this intentionally. But I can tell you it does happen. It's certainly a cause of error that you can see in the data that I analyze for them. I have all sorts of error checking, even at Guide Dogs for the Blind. As pedigree information comes in, I'm checking to make sure that, for example, an animal doesn't wind up in the sire column and in the dam column. I don't know a lot of biology. I know they can do that. So I have some simple checks that I can figure out, and I can go back. I think somebody got recorded wrong, but that's going to throw off everything. And again, so we have to agree that we're going to try and get the best information possible. And then after that, we really do have to cooperate as breeders, and I think that's the larger challenge, is that you need to share this common goal and make use of the information. You need to trust one another that you're going to work together. Because inside a colony like Guide Dogs for the Blind, there's only 1 or 2 people making decisions. They don't have to worry about this cooperation with others. But in a breed club, you're just one part of a larger element. And you want to all move together in some form, but breeders need to be able to trust one another. They need cooperation, drive. I specifically took these from Doug's talk yesterday. Drive and cooperation and willingness to work together and trainability and intelligence, because they were all the words we had for good working dogs. And if we want to be good working animal breeders, I think that's the other element that comes up in some of the breed club work that at least I've noticed from afar. Not being someone who raises dogs in that capacity. I have pets at home, but it's not quite the same. I'm not a member of any club, and so, but having watched it as an outsider, there are times when I can see the tension inside of clubs because some people are not forthcoming with what's happening to their animals. And that's it's only human nature, and there's a shame as if it was somehow your fault, but it's not. And that's really why I put the word trust up there. People have to feel comfortable enough to say, this didn't work out. And we're going to have to work together to try and solve that problem. But if people aren't forthcoming, it's going to be virtually impossible to make this change. So I wanted to leave that as sort of the final bywords of this talk on how you can try and work inside of your breeds. That is, we start thinking about collecting useful information, whether it's phenotypes of interest and the pedigrees associated with them, and be able to work together. Because I still don't think it's a terribly difficult problem. You come to the idea of identifying whom you think are the superior animals, and that can be done without all the statistical mumbo-jumbo. You can just look at the phenotypes and averages of certain sires and some basic simple statistics that you can do in Excel. But you could rank them and say, these are the good animals and these are not. And among the good animals, it's just being able to understand how to calculate an inbreeding coefficient, which I won't do today. But there's software, and if you want, you can write me an email, and I'll be glad to show you the software and how to do it. Because that's what you want to be able to do. If you have two individuals, a candidate sire and a candidate dam, you want to be able to ask what will be the inbreeding coefficient of their kids. And think of all the possible pairings, and then be able to say, I'm going to do this pairing, because they're going to have the kids with the smallest inbreeding coefficient. And that together is, I believe, at least a feasible or workable system for most of you that confined by small-sized populations, but still having some goals and objectives you'd like to do with your animals. And so with that, I'll close and get you ready for lunch. Thank you.